Morning, everyone. Okay, let's get started. Okay, last time we learned how we can use the 3-point test cross to help us to derive a genetic map for 3 genes if they are linked. If they are in LD, right? So if they are on the same chromosome, right? Okay, but at that time we use a diploid organism, the model organism, to help us to do this. So today we're going to learn how we can use Neurospora, which is the haploid model organism, to help us to do genetic dissection. All right. So this is today's main topic. All right. This is the comparison between the haploid versus the diploid. Okay. So for the diploid model organisms, so we can use the famous Mendelian pea plants, or we can use Drosophila. All right. So when we do the test cross, so this is actually the workflow for the test cross, right? So we start from 2 parental lines. So one, so if we are talking about 2 genes linked, so we start from 1 parental line, which is a double homozygous, the capital B, capital A, and the capital B, and the capital B. So another parent will be here. So the lowercase a and lowercase bb. All right. So here we have input genotype. So this is our parental type, the big A, big B, or small a, small b. So we have 2 input genotypes. And then, so by crossing these 2 parental lines, we got a dihybrid F1 progeny, right? So this is the F1 progeny. The genotype of this F1 is like this. And so then we're gonna test the cross, do the test cross using the tester here. So the tester must be the double homozygous recessive, and then we can get the progeny with different genotypes. So this is output from this test cross. As we all know, so if we look into the genotypes for all the progeny, so this is F2 population progeny from the test cross. We're going to see like some of the progeny, the F2 progeny, will carry exactly the same genotypes like 2 parents, right? Like here. So we call this is the parental type, right? But some of the progeny, they show different genotypes when compared to their initial parents. All right, those are the recombinants. All right, so this is for 2 genes. We can extend this to analyze the distance. So yeah, so basically, so if we got these results, so we have learned how we can calculate genetic distance between these two genes, right? A and B. Suppose they are linked, they are on the same chromosome, A and B. So how can we calculate the distance between these two genes? So we have learned, so we can use the recombination frequency, which is defined as the ratio of the number of the recombinants, right? Divided by the total number of the progeny. So this is one way, this is the way we can calculate the genetic distance between these two genes. Now we can extend this to 3-gene mapping. So so this is the 3-point test cross. So I try to give you a very brief review about what we have learned before. So so that was only based on the diploid organism. So today we're going to learn how we can do the same thing in the haploid individuals. All right, haploid organism, the Neurospora. So so the difference is here. So two parents will be haploid, like this. This is one parent, and this is another parent. They are haploid. Okay, and all right, we call their genotype as the input as well. So like the diploid parents here. So we have two genotypes, input genotypes, and then these two parents will form a meiotic diploid myocytes. So this is the term everyone should know. What does the myocyte means, right? So these two parents will mate. They come together to form a transient myocyte. So this is a temporary like diploid cell, and this cell will go through the meiosis to produce 4 progeny, which are haploid individuals. Okay, so if we look into the genotypes of the progeny from this cross, so all the genotypes for the progeny will be named as the output genotypes. Okay, still we can see part of the progeny will carry exactly the same identical genotypes like their parents. Those are the parental type. However, some of them are not. So they are different. They have different genotypes when compared to their input parental genotypes. All right. So the same thing. So the same thing. If these two genes are linked, if they are on the same chromosome, we can use exactly the same formula to calculate the distance between gene A and gene B, right? So what we need to do is we need to figure out which progeny are represent the recombinant, which are the parental type. And once you specify the progeny that carry different genotypes than their parents, then we can use this formula to calculate the distance, genetic distance between these two genes. All right, so, okay. Any questions?
Yes, Professor, I have a question.
Yeah.
So going back to what was it? When you were talking about how these two gametes will come together, form into this meiotic diploid.
You mean this, right? The haploid. All right. Yes.
So going back to that, will this cross in this fashion were to be little a, big A, little a, big B, little b, will this happen every time so that we can get sort of this dispersion of crosses that we get below it, or is that just a high chance of that happening?
So sorry, could you say that again? I don't think I gather your question. So did you?
Well, what I'm saying is, are these two gametes gonna fuse like this every time,
Oh,
and
I see.
or is this just a high chance of happening?
Okay, so this is the difference between the haploid organism versus the diploid organism. So technically, we can only say these are the gametes. We can only say these are gametes because they are diploid organism, right? But for the haploid, they are not gametes. They are actually living individuals. So they only carry one set of the chromosome.
I see.
So these two parents, yeah, that's why we say they are haploid, which means like they only have one set of a genome, right? One set of the chromosomes, right? So they have a very special mating system. So if they want to cross and produce the next generation, so the two haploid parents need to fuse together to form a transient temporary diploid male sites.
Right.
And that, so this is very similar to here. So go through the meiosis and produce 4 progeny, which are also haploid individuals.
Okay, and so then what you're saying is that these green cells right here, right? These temporary myocytes.
Yes.
They're not organisms. They're just that in between step before we actually get to the progeny, which are these yellow and blue cells.
That's right. Yes, that's right.
Okay, got it. Thank you so much.
Sure. Okay. All right. So that's why. So I put this slide here, is because because I don't think we have time to go over this slides here, but this is very important because from now on we're going to use another important model organism, Neurospora, so which is the haploid organism. So please, yeah, read the textbook and understand the life cycle for this guy, and yeah, this will help you to understand the slides. All right. Okay. So, all right. So this is very important. So again, we do not have time to go over this life cycle, but yeah, please read textbook after the class and understand the whole process here. And some very important concepts here. So everyone needs to understand this tetrad, ascus, and the meiocytes. All right. Okay. So why this Neurospora or fungi are so important for in genetics? Because they are as important as the diploid model organism, like the famous pea plants or Drosophila, because they are they have very unique and useful feature that we can leverage in the genetic study. All right, so let's look at this. So okay, oops. So this is the transient, okay, the diploid transient meiocytes. Now this is 2N, right? Okay, so this meiocyte will go through the first meiotic division, so to produce 2 cells, right? And okay, this is the same, like the regular like meiosis. And each of this cells, daughter cell, will go through the second meiotic division to produce another 2, right? Each of them. All right. Okay. Now the interesting thing is like this kind of like meiotic division is not like so they are arranged in a very nice linear fashion. So because this occurs within a sac like this, we call this ascus, and this sac holds this the meiotic product in a very nice linear way. All right. Okay. And what is different from the regular meiosis is like, so this is not the end of the meiosis, because when we talk about the regular meiosis, so one cell will produce 4 gametes, right? But here, this is not the end of the story. So each of this haploid individual will go through another round of the post-meiotic mitotic division. So just reproduce itself. So just make two identical copies for itself. So the final product will be a sac containing like 8 spores for the fungi or for the neurospora. So what's the beauty for this? So we're going to talk about this. All right. So in the exam, let me erase this. It's a little bit messy here. All right. So in the exam, I may give you a question containing the information about this tetrad. So means this a sac or a ascus containing 4 cells, or I may give you a question with the information about a ascus containing 8 spores. They're the same because when I gave you 8, which means these 2 are identical, right? It's equivalent to this one, and these 2 are identical will be equivalent to this one. So I hope everyone will be clear about this. So let's see what's the beauty of this model organism. All right. So this is the regular meiosis in the diploid organism, like the Drosophila. So okay. So this is a regular the cell and 2N cell here, and after the meiosis, so this cell will produce 4 gametes. So now we are talking about diploid organism. So after the meiosis, we have 4 gametes. So they are gametes, not the individual living organism, right? Okay, those are gametes. They cannot survive by itself. They have to join to another gametes to form a zygote. Then it can develop a living organism. So those are gametes. Okay. But those gametes are mixed together, so they are not arranged. They are not arranged in a very nice way. So they are mixed together, moving around. So you cannot distinguish them, right? Because okay, so here let's say we have 2 homologous chromosomes here. So one is this one, the other one is this one, and we also use different color to represent the different centromere for two different homologous chromosomes. So we use the white one to represent the centromere for the first homologous chromosome, and we use the purple one to represent the centromere for the second homologous chromosome. So here I use different color because I want to show you the heterozygosity of the centromere as well. All right. So after the meiosis, we see like 4 gametes here, but because they can move, they mix together. So you can. It is very hard for us to distinguish which centromere is from which parent, right? But when we look at the haploid individual, so like the neurospora, so after the meiosis, the 4 progeny, haploid progeny, will be nicely held together within this ascus, such that we know this half of the progeny will carry 1 type of centromere, and this half of the progeny will carry another type of centromere from another parent. I hope this is clear. All right, so this is a very nice feature because they cannot move, so they are held together, so very nicely arranged in this linear way. So then this gives us a very nice opportunity to study the genetic map by using this haploid model organism. So let me show you. All right. So okay. So because in this diagram, I cannot label the centromere using a different color. So I show you. So this one is the white one, and this one is the purple one. Okay. Now, okay. So now this is the meiosis. All right. The first, the meiotic division. So we see like two daughter cells, right? And each of the daughter cell will go through another round of the meiotic division to produce two progenitors, right? Haploid progeny. If there's no crossing over occurring in this case, so what is the consequence? Let's see. Okay. So if there's no crossing over, so we're gonna see. So in the original diploid myocyte, so we see within the same cell we have two alleles, right? The capital A and the lower A. Okay. Now, if there's no crossing over, so these two alleles are gonna segregate, are gonna separate in the first meiotic division, right? So if there's no crossing over, let me repeat this. This is very important. So if there's no crossing over between these two chromosomes, then these two alleles, big A and small a, is going to separate in the first meiotic division. Do you agree? You see, this big A goes to the one cell and the little a goes to another cell. So they segregate. Okay. And then we call this pattern. So this is the pattern, a M1 pattern, which means the meiotic division 1 pattern. So the reason why we call this is because these two alleles, we are talking about these two alleles, big A and small a, they separate at the first meiotic step, division step. So in this case, you're going to see what? You're going to see this. Because here, as I mentioned, so this is a very special organism, it's a model organism. So this half of the progeny will carry one centromere from one parent. This half of the progeny will carry another centromere from the second parent, right? So if there's no crossing over, we're going to see what? We're going to see the same pattern, like the big A, big A, big A, big A. Or, so this is the octet. So if I put 8 here, it's equivalent to the tetrad here, right? 4. So if I give you 4, this is equivalent to this because each one is going to produce 2 of this. What a messy slice. Let me rephrase this. Okay. Okay, my point is like if this is M1 pattern, and if these two alleles separate at the first meiotic division step, you're going to see this beautiful pattern here. All right, this means there's no crossing over between what? Between this gene and the centromere, right? Because centromere can never change because of this unique feature, nice feature. So if we see the same pattern for this gene, the alleles, the big A, big A, big A, big A, small a, small a, small a, small a, then we say there's no crossing over occurring between the centromere and this gene, right? There's no crossover here. All right, let's look at this another situation. Suppose there is a crossover occurring between this gene and the centromere during this meiosis. So what is the consequence? Let's see what's the results. All right, this is the first division, M1. Did you see the segregation of these two alleles? No, because in each of these daughter cells, you can still see 2 types of alleles, right? So within this cell, there's a big A and there's a small a. Same thing for here, because of the crossing over. So that means like, yeah, so the 2 alleles have not separated at this moment and at this step. So then after the second meiotic division, so what happened? You're going to see the big A separate from the small a, right? Because they end up in different cells. All right, the same thing for here. All right, so in this situation, so why this is different from the previous slides? Because there's a crossing over. So if there's a crossing over occurring between this gene and the centromere. We're talking about the distance between the centromere and the gene, because yeah. But now you can treat the centromere as a gene. So we want to measure the distance between this gene to the centromere, right? Okay, now if there's a crossing over between the centromere and the gene, then the two alleles of this gene, the big A and the small a, is going to fully separate at the second meiotic step division. So we say, if you see this, still like this centromere will not change. They came from one parent. This centromere will not change. They came from another parent. However, you see different pattern for this gene, right? If you see the big A, small a, and the big A, small a, so this is different from the previous slides. Then we say this is a M2 pattern because the big A and small a, they separate at the second meiotic division step. So we use a beautiful name, M2, to represent this pattern. Is that clear? So there are multiple ways to produce the M2 patterns, right? So if this one is crossing over this one, so yeah, if this one crossing over with this one, either way, or if this one cross with this one, if this one cross this one, so it can generate different pattern, but they all show this M2 pattern. All right. So now I hope everyone understands the difference between the M1 pattern, which is big A, small a, or if I give you the tetrad, so it should be like big A, small a, right? So that's the M1 pattern. Now, M2 pattern is a little bit different. M2 pattern will be like this: big A, small a, and another big A, small a. Or if I give you in the question, if I give you a tetrad, so it shows like big A, small a, big A, small a. So if you see this pattern, so that's M2 pattern. Okay. All right. Now the same. So we use the same principle to calculate the distance. Now I hope you are not confused. So here we are measuring the genetic distance between a gene with the centromere. So we want to measure how far is this gene removed from the centromere, right? We can use the same principle. So the distance from the centromere to a locus, to a gene, will be calculated using this very similar, right? We still use a ratio. So previously we learned how we can calculate the recombination frequency using a ratio. Here the same. So the ratio is formed like this. The denominator is the total number of the SI. All right. So we're gonna see how many. So this is one ascus, right? So for example, this ascus is an M2 ascus. M2 pattern ascus. So if you look at another ascus, maybe that's a M1 ascus. So whatever. So you just need the total number of the ascus produced from the test cross, and that's the total number of the SI. So we will put this number in the denominator, and the numerator will be the total number of the SI that have M2 pattern. But if you look into this pattern very carefully, what you can see is, so out of this 8 progeny, or out of this 4 progeny, they're the same, right? So half of them are parental type. Half of them are parental type. So these are parental type. And half of them are the recombinants. Do you agree? All right. So now look at the formula here. So I hope everyone can understand why I put a 1/2 before this numbers, because this numbers represent the S psi that containing the M2 pattern, right? But within each M2 pattern ascus, only 50% of the progeny are the parental type, and 50% of them are the recombinants. We are only interested in the recombinants, because recombinants progeny will represent the crossing over events, right? So now, if you understand this, so this is no problem. So you now understand why we use this formula to calculate the distance between this gene and the centromere. Any questions? Very good.
Yeah, just really quick. Can you define ASCII again? Just for my notes.
You mean this SI? Or how do you
Yeah.
pronounce this? Sometimes, and so this is terrible for me. So how do you pronounce this? SI?
Well, it's just ASCI by the look of it.
All right. So this
But
is
ASCII
the.
is definitely fine.
Oh yes, ascus. Yeah, ascus. Let's see. The ascus means this one ascus. All right.
Oh, okay.
Right. All right. So for example, this one. Let's say this ascus is a M2 pattern, right? Okay. And maybe you can get a ascus like this. This is M1 ascus. Okay. Now, if you do a test cross, you're going to generate maybe 1,000 SI. 1,000 SI with different patterns. So now, so if you have created a 1,000 SI from this test cross, then the total number of the SI will be 1,000. Is that right?
Sure. Yeah.
Yeah. Okay. Now, so from this 1,000 SI, you're gonna look into their patterns to find out how many of them have the M2 pattern. So for example, there's like only 300 out of this 1,000 carry the M2 pattern. All right. So that's the thing we are interested in, right? But if you look at each of this M2 pattern ascus, you're going to see only half of the progeny within this ascus are parental type, and half of them are the recombinants, right?
Sure.
These are the recombinants. So that's why we want to multiply this by 1/2.
Okay.
Because, yes.
And then I guess my second question is and we're trying to figure out specific patterns here. That's what the diagram to the right is.
Yes, right. So yeah, gotcha. Actually, all these patterns are M2, but as you can see, the difference, like, so which chromatids is crossing over with which chromatids, right? So for example, this one cross this one, we're going to generate this M2 pattern. If this one is crossing over with this one, so you're going to generate a little bit different, but you can see they are very similar. So in the test, if I give you some pattern like this, you can immediately pick out if whether they are M1 or M2, right?
Yeah. I get it now. Thank you.
Sure. This is very important. I know it at the beginning it's a little bit tricky, but once you get it, it's very useful. And yeah, believe me, yeah, this is very useful because I'm gonna put questions in your next quiz and also in your next midterm. So this is very important because this is a very nice, most useful model organism that scientists often use for the genetic study. So it's very, very useful. Okay.
Yes, hello, excuse me, Dr. Jia. Just one confirmation. Does the M2 pattern refer to the entirety of the gametes within the ascus, or does it just refer to the recombinants?
Sorry, could you say that again?
Does the M2 pattern refer to all of the gametes, including the parental and the recombinant types, or is it just the recombinant types?
You mean M1?
M2, I mean M2.
M2, yes, okay. So your question is like, so when we look into a M2 pattern, ask us, so what's the percentage of the progeny from this ascus will be parental type, and what's the percentage of progeny will be recombinants, right? So it is, yeah, you're right. It is always like this. 50% of them are parental type, and 50% of them are the recombinants. Yes.
All right, thank you.
Sure. Yeah. Again, this is very important. I hope everyone can understand. Yeah. And this will help you to answer my questions in the quiz and in the midterm nicely. Oops. Okay, so now let me give you an example here. So yeah, by doing this example, so probably, yeah, this can help you better understand the ideas behind using this model organism to do the genetic study. All right. Okay, so if we do a test cross, and this is the progeny, so we have like 1, 2, 3, 4, 5, 6. We have 6 types of SI. All right. So based on this information, you can immediately figure out which one is which, right? So this is so I gave you the tetrad, so rather than the octad. So still, like, based on the pattern, you can easily figure out. So this is M1 pattern, right? Very clear. And this, because the P plus, this is definitely, obviously, this is F2, M2 pattern, right? And the same. So this is also a M2, and this is M1. This is M2, and this is also M2. So it's very easy, right? Okay. Now the question is like, okay, so if I give you this information, how do you determine the genetic distance between this gene, the P gene, and the centromere, right? So it's easy, right? So once you understand what is going on behind, it's so easy for us to use this formula. So in the test, I'm not giving you the formula. So yeah, I'm not giving you the formula. So you have to understand this, and you have to, so when you understand this, you can easily memorize this formula. So the formula is just like this. So the total number of the SI here, so those are the total number. So in the progeny, you're gonna see like 45 SI carrying this genotype, and you got 7 carrying this genotype, and another 7 carrying this genotype, and so on. So that's the total number. You just add up those numbers. So that gives you the number for the denominator, 100. So in the test, I will give you very simple numbers like this. So I don't want you to waste your time on complicated calculation. I only give you simple numbers, so it's easy to calculate. And but you need to show me how you the steps you get this. You need to show me the steps that help you to get your final results here. All right. All right. So that's the total number of the XI, SI, and in the numerator. So as I mentioned, you need to use the total number of the M2 SI multiplied by 1/2, because only half of the progeny within each of the M2 ascus represented the recombinant progeny. All right, and this will give you this. It's very, very easy and similar. All right, and so here, remember, we want to calculate the distance between the centromere and this gene. Okay. Okay, distance. Okay, now, so the second, for example, the second question I could ask, like, okay, so we have like an ascus like this and another ascus like this. Show me what is going on during the meiosis. So this is M1 pattern, so there's no crossing over. So it's easy to get this, right? However, for the second one, this is the M2 pattern. So there's a crossing over occurring between these two homologous chromosomes. So if you can label like this, there's a allele switch between these two chromatids, and you're gonna end up with this phenotypes, right? Okay. Okay. Now, this is the similar thing. So if we still use the Neurospora as a haploid, we can also measure. So just now we learned how we measure the distance between a gene and a centromere, right? So if this time we want to measure the distance between any two genes in regardless of the centromere, so we can do very similar things like we did before in the diploid organisms, okay? So for example, okay, so if we just look at these two genes, okay, A and B, and we do the test cross, and we got different progeny showing and the numbers of different progeny, right? Parental type, and recombinants, and this is the numbers here. So if we got this number, we want to see if these two genes are linked or not. So first, we want to test if these two genes are on the same chromosome or not. So that's one test, right? If the test shows, okay, they are linked, then we can still use this data to help us calculate genetic distance between these two genes if they are linked, right? So I'm gonna just very quickly go over this with you. So you are very familiar with this, right? So if this is the data, okay, so we observed that this many progenies in each category. So if these two genes are unlinked, if they are on different chromosomes, so we're gonna see the independent assortment, just like Mendel 2, right? The law of Mendel 2. And so the expected the expected number of each type of the progeny will be this, and then we can easily construct a chi-square test statistic and use this table to help us to figure out the p-value, and then you can make a test whether these two genes are linked or not. Okay. All right. Now, second, so once we demonstrated that these two genes are linked, they are on the same chromosome, then we have another business, right? So we want to use the data, the test cross data, to help us calculate the distance between these two genes. All right. So remember, this is not the regular diploid organism. This is the Neurospora, so haploid. So we're gonna see the progeny within the ascus, the SI here. All right. So now, so from this test cross, so this is the test cross. All right. And we're going to generate four types of ascus. All right. So the first one, okay, if you look at the first one, either I use the notation like this, or if I put them in this way, so you can easily get, okay, so this are parental type, P1, parent 1, and these are the parent 2. All right, so if there's no crossing over occurring in this meiosis, you're going to generate a ascus like this. So every single progeny within this ascus will be parental type. So if a ascus looks like this is defined as the parental ditype, because we have two types here, and each of these two types are parental type. So parental ditype, and which means there's no recombination, there's no crossover occurring in this meiosis, right? Any questions here? Okay. Now let's look at this one. So if there's a crossover occurring, then you're gonna see what? You're gonna see like, okay, half of them are the parental type and half of them are non-parental, right? So if this is the case, we will give this type as a name as the tetratype, because there are 4 types, right? There are 4 types here. Out of this 4, 2 are parental type and 2 are non-parental type. So this is called the tetratype. And 50% of the spores, the progeny within this ascus are recombinants, because if we want to measure the distance, genetic distance, remember, we are very most interested in the recombinants. So we want to calculate the recombination frequency, right? So this is what we want to calculate. So in this type, only half of them, 50% of the progeny are recombinant. All right, let's look at here. All right, this one, if you got this one, all right, so all of them are non-parental. Yeah, there are many crossing over occurring in this one. So again, so because there are many crossing over occurring in this one, this means a very rare situation. So if you generate a large sample size for this test cross, you're going to see very small numbers, very small number of the progeny of the ascus carrying this genotype, right? Because this is very rare. All right. If it is this type, so it has a name, non-parental type, and all spores, all the progenies within this ascus will be recombinant. Right. Okay. The last one here. All right. So although there's a crossing over occurred, as you can see, so because of the nice feature of the neurospora, so the resultant spores will be held together nicely in a linear fashion. So definitely you're going to see there's a change, right? But this change will not affect the relationship between these 2 genes. Even there's a crossing over, but this crossing over did not occur between these 2 genes, A and B. So if you look at the genotype for these 2 genes, they are all parental type. So still they are parental ditype. Yeah, there's a crossing over, but no crossover occurring between these 2 loci. If we just want to calculate the distance between these 2 loci, so this doesn't contribute. This one, yeah. Okay, now formula, it's very easy, right? So we still use this formula, the basic formula. So the recombination frequency, which is defined as the genetic distance between A and B, gene A and B, is calculated using this ratio. So the denominator is the same, the total number of the progeny, total number of the ascus actually, SI, and the numerator will be the number of the recombinant progeny, right? So in this case, let me erase this. Okay, so in this case, the denominator will be the total number of the SI, and the numerator will be 2 parts. One part is from here. So the tetratype, for tetratype, only 50% of the spores, the progeny, represent the recombinants. So that's why we multiply this number using 50%, right? 1/2. Okay, now another contributor is the non-parental type. And all of the progeny within this non-parental type represent a recombinant. So 100% of them will contribute to this calculation. So now if you understand this, it's easy to understand why we use this formula to calculate the distance between these 2 loci. Is that clear? All right, so this slide just gives you some idea how this happens, right? So if these two genes are linked, A and B, so if there's no crossing over, so this will gives you the parental dye type, right? This is the parental dye type. And if there's many crossing over occurring this, you're going to generate like a recombinants for all this 4 progeny. So this will gives you the non-parental type. And if there's only 1 crossing over occurring, this will give you the tetra type. And for the tetra type, you're going to see like 50% of them are parental type and 50% of the same thing here. All right. Remember the centromere. All right, the centromere will never change. So this is the nice feature for this unique haploid model organism. So this is a very interesting problem. It's a little bit hard. So I hope I'm going to go through this very quickly here, but I hope everyone can come back to this problem again after class, because this will give you a very good exercise to understand the genetic analysis using this Neurospora haploid model organism. All right, so now we do the test cross. So easy, right? Easy. And okay, so from this test cross, we generate a total of 1,000 linear asci. And this is the data. Yeah. So for example, in the test, I can give you a data like this. So if you look at here, so first you want to see if these 2 genes are linked or not, right? Okay. So you see if these 2 genes are not linked, if they are independently assorted during the meiosis, so you will not see this highly unequally distributed data, right? So this will immediately give you a impression, okay, no, these 2 genes must be linked. Otherwise, you're going to see very evenly distributed between these different types of genotype, the aspects, all right? So this is the first impression. All right, they are linked. Then how are these 2 genes are linked, and how are these 2 genes are arranged in terms of related to the centromere. So this is what we want to do now. So first, so if you got this data, so this represented the most frequently seen genotypes for among the progeny asci, right? So what conclusion can you get? This number is significantly larger than the others, right? Which means those are the parental types, because there's no crossing over there. Because the recombination crossing over is a costly event. So that's why if you do a test cross, so majority of the progeny will represent the parental type, no crossing over. This will immediately give you the results. These are the parental type, right? Okay, now once you got the parental type, so you can look each. So suppose, okay, so you can look at. So for example, we can calculate. So the first thing we can do is to calculate the distance between each gene with the centromere, right? So for example, if we look at the first gene, the LIC gene, just leave the AD gene alone. So we just look at the LIC gene. So we can use M1 or M2 pattern to help us to calculate the distance between the LIC gene and the centromere, right? So we have learned that. All right, so if this is the parental type, all right, what is this pattern? This is M1, right? We just look at the LIC gene. We leave the AD gene alone for now. Okay, so how about this? This is still M1 pattern, right? And oh, what happened here? So yeah, this is a little bit different. Okay, then we can put this M2 pattern here, right? Okay, again, this is M2. Easy. M2 and M2. Okay, Let's put 2 here, 2 here. Okay. All right, now we need to calculate the distance between this gene and the centromere. So how do we do that? So we're gonna put the total number here, right? And we're gonna add up all these numbers here. So this one, this one, this one, this one. This is the numbers we need, right? And because in each of this M2 pattern, only half of them, the progeny will be the parental type. So we will need to put a 1/2 here, multiply. We need to put a 1/2 here to multiply this number. So this gives you the genetic distance between this gene and the centromere. Is that clear? It's a little bit complicated here. So today we learned how we calculate the distance between a gene and the centromere, and then we also learned how we can calculate the genetic distance between 2 genes in the haploid neurospora. Right? Any questions here? Okay. Okay, now this is easy. So we can do the same thing, right? So we can calculate the distance between AD gene and the centromere. So this time we're going to look at here. So this is the genotype, the parental genotype. And okay, so this is M1, this is M2, and this is M1 again, M2, M2, and the M2. So that we can do the same thing to calculate the distance between this gene and the centromere, right? The problem is like, yeah, so this is not good enough to construct the final genetic map between these 2 genes and the centromere. There are three possible ways to arrange these 2 genes, right? Either these 2 genes are on different chromosomes, so they are unlinked, like the A, situation A, or if they are linked, they could be like on 2 sides of the centromere, or they may be located on the same sides of the centromere. So based on this data, how do we figure out which one is the right one, is the correct one? Right? Okay, so definitely the first one has been ruled out. Why is that? Because we know, yeah, if they are independent, so we should not be able to see this kind of like highly secured numbers, right? So they should be roughly equal. So the first one will be ruled out, right? And now we need to compare B or C to see which one which one makes more sense. Okay, now, okay, suppose C, let's see, suppose C is correct, all right? So, and when we look at this one, so this one, okay, and so because this is the parental type, oops, this is the parental type, and those are the ASCIIs somehow involving crossing over, right? And if you compare those numbers, this one actually represents a very frequent project from this test cross. Let me remove this. All right. So, okay, so how do we get this progeny from the parental type? So there must be a crossover occurring here, right, to generate this. Yeah, which means, okay, yeah, so if this is true, if C is true, so there's a crossover occurring between the centromere and the NIC, where NIC gene will produce this, all right? However, so if B is true, if these 2 genes are located on 2 sides of the chromosome, then a single crossing over will generate. So this will generate the M2 pattern independently for these 2 genes, right? However, this number is very large, which means they are more likely to to occur, which means like these two genes are very likely linked, tightly linked like this. All right. So this data will give us the results. Okay. This is impossible. And this is the correct mapping situation. All right. And after you figure out this this correct mapping, so we can calculate the genetic distance between these two genes, and also the relationship between this each of these genes with the centromere. So we can use the same strategy to construct the map like this. So yeah, this is a very useful example. It's a little bit difficult, complicated, but I hope you can spend some time after class, and this can help you better understand how we leverage this beautiful, useful organism to help us to do the genetic analysis. Okay, so for this part, I listed some very important concepts here, so they are very useful. Hopefully everyone will understand and know how to use them. If I give you a question like that. So this is very important for Neurospora, a very useful genetic model organism. All right. Okay, so I think I finished this part. So I still have like a little bit time. So let's move on a little bit. Okay, so in the beginning, we start from Mendelian inheritance, right? So at that time, we talk about a single gene, right? We use this classical example, the flower color, the color of the flower of the pea plants, all right? We all know that the big P is the dominant allele, and the small p is the recessive. And if we cross these two parents to generate the heterozygous F1 progeny, and if we do the intercross or we do the selfing, we're going to produce this beautiful 3:1 ratio, right? All right, so this is what we have learned before. Okay, so today we're going to learn a little bit about the modified Mendelian inheritance. So now we talk about a single gene. Next time we're going to talk about more than one genes. We're going to talk about like the interaction between two genes, the relationship between two genes, and how they play together within a biological pathway to affect a trait of interest. So we start from the simple one. So now today we're going to talk about single genes. Single genes, so the dominant one, the basic one is the dominant one. This is the classical one, not modified. And here we talk about the modified, means like the rest of them are a little bit different from the dominant one. All right. The classical flower example, the pea plants flower color example is the basic one, the dominant. So let's talk about the other types of relationship between the alleles for one gene. All right, complete dominance. So this still the basic one. So we have learned. So if for example, if this gene like is a functional one, both alleles are wild type and the dominant. So both of them can produce the functional protein, and this functional protein, the two doses of the functional protein will produce wild type. Easy, right? Okay, so for some reason, like one allele has been mutated. So in this case, this individual will have one mutant allele and one wild type. So this is a heterozygous. The wild type one can produce functional protein, which can do the function in the cell, but the other one, no. So this one is non-functional protein. It can do nothing. But even there's only one dose, it's good enough. So it can also achieve the standard function for the cell. And then what you see is still the wild type. Okay. Only if like this individual has two mutant alleles, then neither of them can produce a functional protein, and then you're gonna see the mutant phenotype. So this is the classical complete dominance situation. So you're gonna see the wild type and the mutant, the ratio is 3 2-1. All right, complete dominance. All right, example. So in human, the skin color, so it's usually like is caused by the melanin. All right, this can give us like the dark skin, the color of the skin, and the skin pigmentation. All right. Okay, so for the wild type, which is the dominant, we use big A to represent this. So if this is a homozygous, a wild type dominant, so if both allele can produce functional enzyme, and this enzyme can get together with the protein to form the functional melanin, then this will give the skin color, some color, the dark color. All right, for the heterozygous individual, if there's only one wild type, the other one is a mutant, so this one cannot produce any functional enzyme, but this one can. And so even there's only one allele, so it can produce enough, sufficient amount of the enzyme that can gives the color to the skin. So, which means like this heterozygous individual will have exactly the same phenotype like the homozygous. So this is the complete dominance model. Okay, only in the individual with homozygous recessive allele, so none of the functional enzyme can be produced, and then you're going to see the albino phenotype. So this is an example. And okay, so incomplete dominance. So sometimes, like some scientists think like incomplete dominance is equivalent to the codominance. So we're going to talk about codominance as well. So actually, the codominance is a special case. Is a special case of the incomplete dominance. So what does incomplete dominance mean? Like, all right. So if this gene gives color to the flower in some plants, all right. So homozygous will give you the purple, and of course the homozygous C1 will not give you any color, so that's white color. However, in the heterozygous, only one allele can produce the purple pigment. The other one gives white, purple, and so because these two the two gene product are both shown, so which means like both phenotypes are demonstrating in this individual. So you're gonna see like there's like purple here and there's white here. So if you mix them together, so they will give you like purple but less purple, right? Some phenotype in between, intermediate phenotypes. So in this case, we call this is an incomplete dominance, and this will be related to another term like the haploinsufficient. So we're going to talk about this a little bit later. All right. So anyhow, so this is the incomplete dominance, and sometimes people say, okay, the codominance. It is also codominance because both phenotypes will be expressed in this heterozygous individual. Then you're going to see a little bit different phenotypes for the heterozygous individual when compared to the homozygous dominant individual. All right. I think I'm gonna stop here today. We're gonna continue this next week. I'm gonna leave a few minutes here in case you have questions. Yeah, I would be happy to answer your questions if you have some.
Hello, Dr. G. I had a question regarding the earlier solved problem for Neurospora.
Yeah.
I'm trying to understand why the answer is C instead of B.
Sure.
So is it because the NIC and the AD genes are on the same side of the centromere, while in B they're on opposite sides?
Yes, yeah, I think you're right. So, okay, so let's try to do the reasoning to rule out this possibility. Okay. All right. So suppose this is true, right? Okay. Now, so from this parental type to this. All right. So suppose this is true. So let's look at the leak gene. Okay. So if you want to generate this pattern from here, that means there's a crossing over occurring here, right?
Yes.
Okay. Then this will gives you what? Gives you the leak plus 80 because 80 doesn't change, right?
Right.
Yes, and this is the leak. Let me see, 80, right? And let's see, leak plus 80 plus leak. Let me see. Yeah. So there's a change. Okay, change between this. Yes. Okay. So this will be 80. Right? Okay. Now, if this is true, if there's no crossing over here, but a crossing over here, so you're going to generate what? Leak plus 80 leak plus 80 plus, right? Leak, oops, 80 leak 80 plus, right? Is that right? So do we have this pattern
We
here?
have the left pattern.
Let me see if we have this kind of. We actually do not have this pattern, right? Let me see. Do we have this pattern? Leak, 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 leak plus, leak plus, leak, leak, and 80, 80 plus, 80, 80 plus, leak, leak. We do not have this, which means this is very rare, right? Do you agree? So we do not. Can you help me to identify if we have this pattern here? No.
Yes, I don't think we have that.
Oh, okay. So if this is true, you should be roughly see. We should be able to roughly see the same progeny of this and this, right? Because it's roughly so. Even there's the distance between these two genes to the centromere is not identical, but roughly, yeah, they have equally chance to have a crossing over between here and here. Then you're gonna see this and this with roughly the same numbers, but you do not see this. What does that mean? That means, yeah, this is not impossible. This is impossible, right?
So is it because of the specific order of the NIC and the AD that option B is not possible?
Yeah, because based on the data, I mean, usually like, so before we make any conclusion, we could have different options, but if given the data, so data will help you to identify the correct conclusion, right? Does that make sense? Oh, no. I think, let me see. Oh, no, sorry. Yeah, we do not have this pattern, right? Is this the pattern? No, this is not the pattern. Okay, so this is not the pattern. Plus, right. This is not the pattern. Right? Is that clear? Not very clear. Okay, let me repeat this. Let me repeat this. Sorry. Erase this a little bit. Okay. So sometimes if we want to rule out some possibilities, so we can assume if this is true, can this assumption yield this number, this data, right? So this is one way we can say. So if given this is true, if the possibility that we generate this data from the test cross is 0, is very low, then we're gonna rule out this possibility. Is that right?
Sorry, could you repeat that?
Sure. Okay, no problem. Second. So sometimes, so when we want to rule out one possibility, so we can first assume this possibility is correct, right?
Okay.
All right, given this is correct, so we're gonna see if this possibility can generate a data like this. If given this is true, it is impossible to generate a data like this, then we're gonna rule out this one, right?
All right.
Okay. So now, so let me give you an extreme example here. So suppose this is a centromere, and the genes are on both sides, and they are of the same distance. So they are equal. They are equal. I mean, equally removed. So the distance between each of these genes are, let's say they have the same distance. There is the same distance, like 10 map unit between this gene to the centromere, and the same. There's another 10 map unit between this gene with the centromere. So I'm going to give you this extreme example. Okay. All right, then. So the crossing over, the probability having a crossover between this gene and the centromere and this gene and the centromere should be equally, right? Should be equal.
Right.
Okay. Now, okay. So, all right. So if this is true, let's say, so if there's a crossing over occurring here, then this will generate from this parental type. This will generate what pattern for you? Can you tell me? If there's a crossing over between AD gene and the C, so AD gene and the C, this will generate, because there's no change between the leak and the C, right? So the leak and the C will be the same. Leak plus, I'm gonna use 4 rather than 8, because leak plus, right? And the leak. This is for leak because there's no change between leak and the centromere. Do you agree?
It'll generate a tetratype for the.
Right. So I mean, if there's only one crossover occurring between AD and C, so there's no change here, right, in this region.
Right.
So you're gonna see the same pattern, like the parental type here, right? I use 4, means like leak plus, leak and leak. This is the resultant pattern for leak, right? Because there's no change here. But there's a change in AD. This AD, we're gonna see AD and AD plus. Let's say you use this. And AD plus, right?
Right.
Okay, all right. So this is one possibility. Okay, since the chance to have a crossover here is equally likely to have a crossover in this region because we set up this situation, right?
The probability of the double crossover happening.
No. So this time, so we do not have this crossing over. Let's say we have a single crossing over here. And this should have the same probability, like a single crossing over in here, right?
Right.
Okay, let's see what happened here. So if this is the case, we only have a crossing over here. So what's the results? So this time AD will not be changed. So we're gonna see AD and AD plus and AD plus, right?
Right.
But leak, there's a change in leak plus, leak plus, and leak, right?
Right.
Okay. Now, so if this is the case, so you're gonna see from this cross, you're gonna see equivalent number of the progeny for these two types, right?
Yes.
Okay. Okay. Now, because so this situation is a extreme situation, all right, and it's different from here if this is true, because we have less distance between the leak gene and the centromere, but longer distance between the AD and the centromere. But still, like we can say, like they have roughly the same probability to have a single crossing over in these two regions, right?
Yes.
Okay. Now, so we should be able to see if this is correct. We should be able to see this have some numbers and this have some numbers, and they are roughly the same. But in the data, you only see this. You do not see the other option.
I see. So because we do not see any of the other Nick plus AD, Nick plus AD plus, Nick
Mhm.
and AD and Nick AD plus,
Mhm.
it means that there cannot that there has to be
Right. The B is impossible. So because this result is from the assumption that B is correct, right?
right.
Yeah.
Right, I see. In situation B, it would be a bit more distributed evenly, but since we don't see that pattern, it has to be C. We also know that it can't be A because
Right.
it's
Because
established there is a
this
link.
number, this number is too large. So we cannot believe like they are independent assorted.
Okay, so if I understand it correctly, then the problem with solution B is similar to solution A, since in situation B, even though there is linkage, we should still expect to see some results of the AD undergoing a tetratype.
Mm-hmm. Yeah. So if the same reasoning for ruling out A, because if these two genes are not linked, if they are on different chromosomes, so we should be able to see, but we should not be able to see like one type of the ASCUS having so many represented has this large number. I mean, so you should be able to see like all the categories should roughly have the same numbers.
Right. The Nick and the AD are in option C. They're more likely to be inherited together.
Mm-hmm. Yep.
All right, I understand now. Thank you very much for your time and help.
Sure. Yeah, This is I'm very happy that I can be helpful. Yeah, this is a very good exercise. So once you understand this, I think the problem shown on your quiz and the midterm should be easier than this one.
Yes, thank you very much. I look forward to applying these concepts.
Sure. Good luck.
And you have a nice rest of your day.
You too. Bye-bye, everyone.
Goodbye.